Prueba de sonido, comité, ¿qué tal se escucha?
Se escucha,
Bien,
Pablo.
Pablo.
Imagen.
También.
Dr. Frank, can you hear me?
Yes, I can hear you. Can you hear? How is my sound?
Pretty good. We can hear you very fluid.
Right.
We're about to start in like about 5 minutes.
Okay.
We are going to present a speech from our director of studies, and then we can go with your presentation.
Okay. I don't know if my picture looks bright enough.
Yeah, we can see your camera very well.
Okay.
Thanks for waiting.
You've been sitting in the bright lights all week.
Yeah. I'm going with the screen of loading. Doctora Marcela, ¿nos escucha?
Sí, te escucho, Pablo.
Ok. Vamos a comenzar como en unos 5 minutos más.
Ok.
Gracias por esperar.
Vamos a la cámara.
Público y autoridades presentes, tengan todos muy buenas tardes. Antes de dar comienzo a nuestra charla de clausura del primer Congreso Virtual de Estudiantes de Bioquímica, queremos invitar a la doctora Marcela Wilkens, jefa de carrera de Bioquímica de la Universidad de Santiago de Chile, a compartir algunas palabras.
Estimadas autoridades presentes, estimada comisión organizadora del primer Congreso Virtual de Bioquímica, colegas y estudiantes, estimados alumnos, el Congreso de la NEP se ha realizado todos los años en alguna sede universitaria donde se imparte la carrera. Reúne cada vez más jóvenes con un interés común en la ciencia. Este año, en la situación de pandemia, no era posible realizar el Congreso. Hace 2 años atrás se formó un grupo de estudiantes que fueron los encargados de organizar el Congreso en la USACH. En ese año enfrentaron muchísimas dificultades, logrando como resultado un evento con mucho éxito. Este mismo grupo y otros estudiantes que se sumaron hace unos meses atrás no aceptaron un año sin Congreso. Decidieron emprender este nuevo desafío en la situación de crisis sanitaria en la que nos encontramos. Un desafío esta vez mayor, pues no había experiencia en un evento virtual. Cuando informaron de esta idea a las autoridades de la facultad, supuse que sería un par de conferencias y alguna que otra actividad. No imaginé un programa de congreso con todas las actividades de un congreso normal, con conferencias de científicos internacionales reconocidos, participación de muchos científicos de varias universidades nacionales, charlas, foros, talleres, cafés con un doc. De verdad, un congreso tal como han sido en los años anteriores y en condiciones normales. Reunieron en estos días a muchas personas talentosas en diferentes áreas de la ciencia y así muchísimos jóvenes pudieron participar de la transmisión del conocimiento y entusiasmo por quienes hacen ciencia. Otra sorpresa fue el interés de los socios ANEP y muchos otros no socios en participar en este congreso, con un alto número de inscritos. Creo que también la comisión organizadora se sorprendió por el resultado y de su trabajo y así han sido compensados. Y este resultado que aún no finaliza hoy y mañana creo con un juego virtual, les demuestra a todos ustedes que con esfuerzo y voluntad se puede hacer lo que se proponga. También es admirable que estos jóvenes hayan considerado unir la ciencia con una acción social, ya que muchas familias que ahora la están pasando muy mal han recibido una pequeña ayuda de parte de ustedes. Felicito a la comisión organizadora, porque en poco tiempo logró reunir a tantas personas jóvenes y no tan jóvenes, estudiantes, a profesionales, interesados y apasionados en el quehacer científico. No podía un virus dejar a tantos sin su congreso. Siento un gran orgullo por ustedes. Han mostrado con creces que las dificultades no los amedrentan, se aventuran a grandes desafíos y sobre todo muestran un gran interés por la ciencia. Gracias.
Le damos las gracias a la doctora Marcela Wilkens por sus palabras. Les queremos agradecer a todas las personas que realizaron un aporte solidario a través de nuestra campaña de beneficencia, enfocada en ayuda directa a personas en situación afectadas por la pandemia en el Bosque, Peñarolén y Puente Alto. Para comunicar el cómputo, invitamos a nuestra delegada NEP y miembro del comité organizador, nuestra jefa Patricia Romo.
Es difícil hablar después de tan bonitas palabras de nuestra jefa de carrera en realidad. Estoy un poco con sentimientos. Pero es verdad, en esta actividad no solo queríamos difundir ciencia, queríamos ayudar a las familias. Y me alegra comunicarles de parte de la comisión organizadora gracias a todos los que nos ayudaron y decirles que gracias a su ayuda pudimos lograr juntar 665.000 pesos que podrán ir en ayuda de todas estas familias. Así que muchas gracias a todos los que pudieron donarnos.
Agradecemos la colaboración de nuestra delegada, Patricia Romo, y la colaboración de todos los los partícipes de esta campaña. Pronto estaremos en contacto con las agrupaciones de ayuda humanitaria para transferir esta ayuda. Deseamos indicar que nuestra siguiente charla será llevada a cabo en inglés. Es un agrado presentar en esta charla de clausura al Dr. Frank Robb, bachiller en ciencias con especialidad en zoología y fisiología de la Universidad de Ciudad del Cabo, Sudáfrica, y doctor en biología de la Universidad de California, Estados Unidos. Con una amplia trayectoria docente e investigadora desde hace ya más de 50 años, el Dr. Robb es una eminencia dentro del campo de la astrobiología y microbiología de organismos extremófilos, con más de un centenar de publicaciones y trabajos asociados. Ha sido condecorado con membresía por diferentes organizaciones y sociedades científicas, tales como la Academia Americana de Microbiología, Asociación Americana por el Avance de la Ciencia, la Royal Society of South Africa, la Sociedad Sudafricana de Microbiología, entre otras. Actualmente es académico de la Universidad de Maryland, Baltimore, en el Departamento de Microbiología e Inmunología, donde sus investigaciones sobre proteínas de shock térmico o chaperones le han conducido al mejoramiento de de procesos industriales y biomédicos, tales como la búsqueda de mejoras en la durabilidad e inmunogenicidad de vacunas vivas, lo que mejoraría el almacenamiento en países en desarrollo que carecen de refrigeración. Esta tarde nos presentará su charla titulada Adventure in Extremophile Hunting. Le damos una bienvenida. You can start whenever you want, Dr. Frank.
Thank you. Can you hear me, Pablo, and everyone?
We can hear you a little low.
Okay, let me talk a little higher. How is that?
Yeah, it's better.
Then I share. So how do the slides look? They're up there.
We can see it pretty good.
Okay. So buenas tardes. We are together in a sense, and I just want to say thank you very much for the people who brought together this opportunity to meet in this rather strange circumstance of ours. I want to thank Felipe and Pablo, who've helped me in getting prepared online. I thank Jenny Blamey, who was, I think, instrumental in this, and just thank you, Lord, for giving us the chance to be together. So my title today is Adventures with Extremophiles. I'm kind of in the end of my time in the lab and in my science career, so I can look back on many adventures, so I'll take you briefly through some of those. The plan for the talk is that we talk about some of the physical and physiological tests that extremophiles must undergo, and somehow the research also demands those type of tests. On the front page here, you can see that my position here is at a marine and environmental institute, but also in the medical school of medicine in the Department of Microbiology and Immunology. So my research somehow is fitted into those two niches. Now, a little word about extremophiles. They must go into very, very strong challenges beyond which we would think life could survive. So, for example, this is a sulfatara, pure sulfuric acid, pH 0.5, temperature up to 100 degrees, and yet there are still organisms you can find in there. So they are extraordinarily adapted to stay in these environments. The hypothesis goes that actually wherever there is water on Earth, the possibility exists for life. So we're talking about highly salty on the left, highly acid at the lower left here, frozen with tiny little rivers, and also in the hydrothermal vents well above the boiling point of water, and also down in the terrestrial hot springs we see competition with many different types of organisms, all fighting for a place. We talk a little bit about the mechanics of going to find these organisms. Over the years, my lab has done the exercises of going out into the environment to collect samples which have strange microorganisms in them. And so here is some of the extremophiles from my laboratory over the years. These three pictures are from Iceland. So I think, maybe I'm wrong, but this might be the time that I met Professor Jennifer Blaney. Then some more aspects of this. First of all, we have the waterborne assault on the extremophiles. That's me. And here are the two scuba divers who are still close friends of mine. And we had time in the lab to do the samples. And then the other thing is we have a long history and the Siberian volcanic fields. This is the Uzan caldera, and pretty much my favorite place. And here you see from the slope of the rim of the volcano, the slant of it, it's a volcano which is about maybe 14 kilometers across in diameter. And you're sitting here in the middle of the caldera, the Uzan caldera, in a really wonderful environment for all sorts of extremophiles. Here's Steve Techman, one of my last graduate students who was collecting samples at these thermal fields. You can see he's wearing these boots which protect you if you should fall through and get into the mud, into the boiling mud. This is also on the Kamchatka Peninsula. It's Mount Mudnovsky, an active volcano in the background there, and it supplies electrical power through geothermal energy to the most people in Kamchatka. So there's lots and lots of exploration still remaining to be done, and many, many, many more organisms to discover. And so it needs time to get together with colleagues from all over the world. And that's why these congresses are so favorable now, because we can get together without actually physically traveling. This is me finally in the Shinkai 6500. It's a submersible from Japan. And so colleagues from Japan have been very, very instrumental in helping me doing things that I couldn't do myself. For example, getting samples from the deep sea. Okay, so the extremophiles themselves, the adventure is collecting them, but there's also an adventure. Every time you get an extremophile strain out of the fridge, you don't know, out of the freezer, you don't know what you're going to get. It can be that the extreme that they're at is approaching the limit. And here are some of the limits that the records are being set. For example, halophiles wanting to grow in salt, up to saturating. Thermophiles, the limit here is 121 degrees, which is above the autoclave temperature. Psychrophiles, the minimum, in other words, the record low temperature is -12 degrees. That's also in very salty condition because otherwise would freeze. And piezophiles, able to withstand high pressure, can go up to 1000 atmospheres. So within these ranges, life is bounded as we know it. Maybe these can be extended, we don't know. And that's one of the romantic challenges for extremophiles. The other romantic challenge is we don't know where they classify in the spread of biodiversity. Way back in 1977, Carl Woese, the late Carl Woese, discovered that actually there were, despite what it said in the encyclopedias and textbooks, that there were 3 domains of life. And this has been proven correct: bacteria, the archaea, and the eukarya. So here's humans here. We only form a very small part of the diversity that's in the rest of the microbial world. This tree has been argued about to a huge degree, and we don't know what its shape is actually. More recently, this tree of the archaea, which is shown separate from the bacteria, is now, of course, they are separate, used to be called archaebacteria, is shown here. There are many branches here which are thermophile, mostly extreme thermophile. So it looked like the tree would branch from a thermophile or hyperthermophile. It seems like this is not standing up to the test of time. Nowadays, people have rewritten this tree as follows. This is a tree based on many uncultured species. Everywhere you see a red dot is a lineage, a whole big group of organisms which don't have a single cultured representative. And so what we see here are the bacteria, a huge diversity of bacteria, much more than we suspected back in 1977. So there are many millions of taxa of bacteria. But here we see, very interestingly, the eukaryotes, which is us, coming down here somewhere, in the green, and the archaea are belonging to the same lineage. And some lineages of the archaea, which are uncultivated, are actually on the very same branch that the eukarya are coming out of. So this is controversial. It says that, the hypothesis says that actually the archaea and the eukarya were phylogenetically joined, and that there was a common ancestor of eukarya and archaea somewhere around here. And actually the new lineages, which are now the deepest branches of this tree, are being found to be uncultured, and they actually have not so many thermophiles. They mostly are growing at cold, deep sea temperatures. So that's, and they also have many different eukaryotic properties. So let's talk about high temperature organisms. It's where I've spent most of my life exploring. Here you take a little bit of for consideration here, where we say the hyperthermophiles, which are limiting life at the upper temperature. Maybe they have the key to where life evolved, maybe not. They certainly have many of them are living deep in the subsurface, and they point towards potential for life in extraterrestrial environments. Many organisms use ferric iron, Fe3, as electron acceptor, and this is very common in the deep biosphere. So, and the cultured microorganisms are many of them called as some of the most extreme organisms. The previous record holder, strain 121, was growing on formators an electron donor and ferric ion as an electron acceptor. And it grew in the autoclave. Nowadays, however, we know that there's a new field. The field is actually bounded by organisms that grow like this one, Pyrococcus furiosus, kind of the lab rat, the experimental mouse of the Archaea. This organism at 100 degrees, growing optimally around 100 degrees centigrade, has now been published about maybe 600 or 700 papers with the name Pyrococcus. So this is a very productive organism in terms of science. Talk a little bit about the methanogens. One of the things that happens nowadays, of course, is global warming. And one of the most potent greenhouse gases is methane, biogas from landfills, and all other sources around 900 million tons a year. And of these tons, most of them are caused by methanogens growing. Methanogens are Archaea, but they're also the most versatile Archaea because they grow all across the temperature. There you see rice paddies, and you see many Archaea are living in the rumen of cattle. You can see great emissions coming out of cattle if you could visualize them. Methanogens are growing from, it says 100 degrees here. I'll show you how that looks, down to cycrophiles from Antarctica growing below 15 degrees centigrade. So they are extremely versatile. One of the interesting things that they were quite well known, people would find that you could light gas coming out of rotting pools. We'll try and see if this works. This is a demonstration of what happens when you light the gas coming out of the methanogens in the deep. And so it can be lit, it can be used for that brings us to the highest temperature organism currently, an organism growing at 122 degrees. And this is a strain that's been published and has been around for a while. Strain 116, and isolated from the Indian Ocean. And this strain actually grew at 116 degrees when first isolated, and was from a deep sea smoker, and therefore growing under pressure, it was exposed to very high temperature, well above boiling point of water. So this strain grows to 116 degrees maximum temperature at 5 atmospheres. However, at its isolation pressure, where it was under the ocean, of 40 megapascals, the growth range is extended to 122 degrees. The viable cells were detected in incubation at 130 degrees for 3 hours at high hydrostatic pressure. Interestingly, the methane that's formed is isotopically enriched for heavy carbon and deuterium. And that revises the geochemistry textbook dogma, because the textbook dogma says that isotopically light methane is from microorganisms, and geothermal methane is isotopically heavy. But this is the wrong way around. People have no explanation for this right now. The way this was done is very novel, very simple, and ingenious. To grow and count these organisms, you need to grow them in pressure. And so what they advised was these syringes, which have the ability to put into pressure. You put the organisms inside here, you put them in their pressure cylinder, and then pump the cylinder to a high pressure, and this allows them to grow at a high temperature, and then you take them out and then proceed to count the ones that grew, and from that you can count the number of cells that are growing and get a growth curve. And with this and these type of apparatus, they were able to, with a lot of work, establish a good data showing the growth curve at above 120 degrees. We also, for large-scale growth, have fermenters. Here's the fermenter at our lab at the IMET. This is the fermentation in extreme environments laboratory, Fermexen laboratory, and it's a really a replica of hydrothermal vent to bring the pressures and temperatures and the gases to in line with what's in the deep sea. Maybe I'll show you this. Hope this works. I'm going to stop sharing and then share again.
Dr. Frank, you can try to share screen again.
Yes, I'm trying to. Yes, here we go. So this is a more alive view of the. You'll all feel a little seasick after a while, which is also gives you some feeling for what's happening in the deep sea research. So this is what the inside of a extremophile fermenter lab looks like. Okay. We'll go back to the slideshow here. Okay, and this is a picture of Carl Stetter. Actually, he is one of the pioneers of this field. He really was the first one to successfully cultivate hyperthermophiles growing above 100 degrees consistently. Here he's shown. He looks a little fanatical. It's a true look. He's standing next to his fermenter, and the fermenter is interesting because it's a titanium fermenter. I'll show you the picture here, which is actually the aircraft called the Blackbird, SR-5 Blackbird aeroplane. The fermenter and the Blackbird aeroplane have one thing in common. They're both made out of titanium. Carl Stetter found out that the highest temperature organisms would not grow when they were transferred from the glass vessels into stainless steel. And one metal that they could do successfully was titanium. So he got a lot of money from the German government and had them buy a fermenter from the Swiss engineering made out of titanium. So not everybody can do that. A little bit about how organisms can do these extraordinary things. Proteins that they have to rely on are packed structures. And here is a picture of a breakwater in Israel that made me think about what is happening inside the structure of a protein. These elements here are packed together. Each one element is quite flexible and movable. However, when they're packed together, they make quite a rigid wall here. And so the proteins that the hyperthermophiles have something like this. They have extremely effective packing. However, if they pack too tight, then the activity of the enzyme or the flexibility of the protein to interact with other proteins is interfered with. So it's important for them to actually have just the right amount of flexibility. And so it's always been a fascination for me how this has worked out. Protein stabilization, the way proteins get to be less or more stable, is as follows. There's increased packing of the hydrophobic core of the protein. Most proteins have hydrophobic inside and hydrophilic surface. The decreased length of the surface loops, the replacement of the loops with helices, alpha helices, and then the immobilization of the amino and carboxy termini, which are buried in the molecule. And then what we found historically, way back in the early days, is that the increased number of ion pairs or salt bridges was a feature of hyperthermophilic protein. And this, we found out, was a feature that from our model structure, this is a picture of the skeleton of the glutamate dehydrogenase enzyme from Pyrococcus furiosus, the 100-degree organism. And what you see here are wherever there's an arrow with two heads, it's a negative and a positive amino acid going together. And what you see within the molecule as a skeleton, like a skeleton, there are ion pairs, and here there were actually 16 ion pairs in a network, in a continuous network of charge, buried in the molecule, buried inside the protein, and engaging different subunits of the protein. And so this ionic network is a feature of very high temperature protein. Then whatever happens, usually proteins are destabilized by pressure. What we know is that many of the organisms that grow at high temperature, if they grow above the boiling point of water, then they normally have a high pressure. Proteins are normally destabilized by pressure. They fall apart above, say, above 200 atmospheres. However, we found with the glutamate dehydrogenase, that same enzyme, that if we increase the pressure from 5 atmospheres to 500 atmospheres, that the stability, this is the half-life described here, went from 14 minutes at 5 atmospheres to 310 minutes at 500 atmospheres. So that was 22-fold increase, 22 times increase in stability. So that was unusual, but you can imagine it happening in the organisms in a systematic way, where these proteins are responding to pressure and being kept intact and more stable. My graduate student, one of them, thought maybe we should try out some of the proteins from extreme thermophiles to put in E. coli. So what this slide is colonies of E. coli shown on plates. And what happened was that he put a heat shock protein into the E. coli as a recombinant protein. And then he asked what happened if you took the plate and put it at 50 degrees and then left it to survive what she showed was actually that this E. coli strain that had the heat shock protein from the hypothermophile was much more survivable overnight than the one that didn't have. So it looked like this was creating a thermophile from one protein. So I was very skeptical, but we went on. We tried this again. We could actually show here, starting out over 120 minutes and measuring the number of viable cells quantitatively, we found that after 2 hours at 50 degrees, we could find out that the survivors with the small heat shock protein compared with the control were about 16-fold more. So there was a survival advantage by having just that one protein. This was kind of interesting to us because we actually still don't fully understand the mechanism for this. But there it is, it works, and the heat shock proteins, the chaperones from these organisms, actually help E. coli. Not only help stay alive, but they also help E. coli grow at 45 degrees, shown here with the small heat shock protein compared with the control without. So they were able to keep the E. coli growing at temperatures beyond where they would normally stop growing and even start to die. So from that finding, we, just about that time, I transferred to the medical school, and it became important for me to do some work which was medically related. And I thought of maybe adapting that phenomenon, being able to enhance the stability of bacteria. This could be applied to bacterial vaccine to make a connection. Now, how to use extremophiles in vaccines. The concept was to use the adaptive protein salvage mechanism, that's the heat shock protein, to stabilize vaccine for long-term storage and transport into the developing world, where there are countries that don't have refrigeration, they don't have cold chain to keep their vaccines. And so these vaccines would be able to stay long periods of time at room temperature without destabilizing. So here's what we did basically. We started with an organism which is used as a typhoid vaccine for the typhoid fever, Salmonella typhi. It's actually an attenuated strain of the one that gives you the disease. You drink it, it goes into your stomach and into your bloodstream. But because it's attenuated, it's very delicate, fragile. It has to be kept in the refrigerated condition. What we did was to take these extremophile chaperones, which are heat shock proteins which can salvage proteins inside. We put them into the salmonella, and then we looked at what would happen to help the salmonella survive. Here's the basic experiment we did with E. coli, surviving E. coli at 37 degrees with very difficult conditions, actually dehydrating them. We find that with the chaperone, that they're after 8 days surviving, whereas the ones without the chaperone are dead. So that was part of the positive results. We ended up trying all of the different chaperones that we had in the laboratory, which was amongst these ones, to find out what they could do. And each of them were tested for increasing the viability of E. coli. So here is the viability increase. The one that we described already was the Pyrococcus furiosus small heat shock protein. It was increasing viability for 16 times. However, we find that, interesting, we found some, the chaperone had no effect, whereas some had a 400-fold effect. And here, this particular one, the E. coli could be stored after 2 hours at 50 degrees, and they had no loss of viability. So they had extraordinary stabilization because of this. It happens that this organism is a psychrophile growing optimally around 20 degrees. It's from Antarctica. So it's, for some reason, its heat shock protein is very, very efficient at detecting E. coli. So then we tried that in the Salmonella typhi, and indeed we could find a 600 times increase in stability for that. And we then went back to the same type of experiment with incubation, suspended it, and could find out that as we were suspending it in that sort of condition, we could look at the expression of green fluorescent protein in these strains. And the control strains are shown here. And this is a proteins visualized on a gel, and by fluorescence, it's autofluorescence. So they are fluorescent here, but what you see here is the expression of the chaperones, the MB chaperone and another chaperone which was successful, was enhanced, so it was actually keeping these green fluorescent proteins alive. We also took the capsule protein from the plague organism. Yersinia pestis causes the black plague, and the capsule protein, the actual surface protein on the cells, is what is engaging with the immune system. So when you find a vaccine for plague, you have to have immunization against this capsule protein. So what we did was to express the capsule protein in the strains that we made. First of all, in the strain here with no chaperone, here's the strain without any of the capsule protein, F1 capsule protein. Here's the strain without any chaperone, and here's the strain at the end with chaperone and with the F1. And what you see is that there's more, and it's highly polymerized to form the surface of the protein. So what we did was to actually make a double valent vaccine, bivalent vaccine, which would go after typhoid and plague at the same time. Unfortunately, the Gates Foundation is not really interested in typhoid, in plague. It led to a more interest in the chaperones that are in archaea, and we look here at the whole proteostasis, the whole network that keeps proteins stable. Archaea have a simplified system, including just a few components. These components that are out of focus are the ones in eukaryotes that archaea don't have. So we could focus in these ones in their function and look at them in terms of how they could contribute. Molecular chaperones, many of which are heat shock proteins, are really in two big classes. They're either holdase, which means that they hold the unstabilized proteins, they hold them together and don't allow them to precipitate and fall apart, or else they fold them. They're called foldases. So these actually actively fold the proteins and bring them back to their native conformation. And so there are two sorts, and they work together. It's like a constant. Here's an example. Chaperones are large cylindrical structures. These are 1 million molecular weight, and they have, as shown here in this visualization, they have an internal cavity into which the unfolded protein goes and is folded. The way they get to there is the interaction between pre-folding which is a whole day chaperone that now holds onto a protein and then interacts with the chaperone and delivers the target protein into the cavity chaperone. Here's a simplified version, and it shows first of all, here's the prefoldant. Here is the chaperone from humans, for example, or any other eukaryote. They have a very big complexity. They have 8 subunits in one ring, and the same 8 subunits in another ring. And so the thing is, the whole structure is 16 subunits with 8 different polypeptides. If we look at Pyrococcus, it has 16 subunits, but they're all the same subunit. They all are the same gene. And to express Pyrococcus chaperone, you just need one protein to express an eukaryote. And so that's an idea of what has happened between the complexity of the archaea and the complexity of the eukaryote. Eukaryotes have got 8 different subunits that are generating and all doing similar jobs with having a bigger diversity. The mechanism for this is shown here. Basically, here is what happens in bacteria and in the organelles of eukaryote. You have a cavity, and then you have a unfolded protein showing here in the light blue entering, and then you have another protein coming on top and being put in place by the binding of 7 ATP molecules. And then as the ATP molecules are consumed, in other words, they get turned into ADP, you get the formation of conditions inside this cavity which cause the proteins to fold. That's to say they turn into blue. And then as the ATP comes in to replace ADP, then that is discharged, because that opens the cavity and the protein goes away. In the second group of these, there is no separate lid. There's a little apical domain here, shown by these little green things at the end. And these are what interacts with the protein or else with the prefolding in order to get the protein inside. In many other ways, the cycle is very similar. Binding of 8 ATP molecules, 1 per subunit, and then binding of ADP, and then release. So that cycle is complex, and you can imagine what's happening in these molecules. They have hinges, the subunits, that allow them to open up. And shown here is some of the first quick images we got of a chaperone, and was done by electron microscopy. In the early days, we could just barely see that, first of all, that there were 8 subunits in the top view, and from the side view, we could see that there was a cavity and that there was a closure at the top. So this subunit is actually carrying out that particular built-in lid function. Nowadays, we have a structure. There are many structures of this, and we did one structure which behaved very beautifully, showing the open and closed forms by either binding ADP, in which case we had this form in the purple, or AMP PMP, which is an ATP molecule that cannot be transformed, so it's unhydrolyzable, and so it fools the complex into thinking that ATP is bound. So that opens it. So here are the 2 pivots. The thing opens as a concert. It's like a flower opening, and it's helped by hydrolysis of the ATP in this core position. And we did a lot of mutations here to confirm the functions of these where the whole complex has to sense the presence of ATP by these residues, basic amino acid residues. And so we see here the open form and the closed form. You see there's a pivot here. It's like a ball and socket joint, like a hip joint in the human skeleton. Very interesting. Won't go into here, we're short of time, but here is what's happening as a movie. Side view showing the amount of movement that happens in this complex. Now bear in mind, this is a complex that can go up to functioning at 90 degrees centigrade. And this has to happen at 90 degrees centigrade. And here's what it looks like from the top. This very beautiful structure was made by our collaborators who did the crystal structure, the Sunshin Cha lab, and his students and his wife who works in the lab. They have a really good method for converting the open and closed structures into a systematic movie, which shows much detail as to how the thing actually works. It also allowed us to look at the size and the internal charges on the cavity, showing here the cavity with much negative and positive charges to force the hydrophobic skin of the unfolded protein to internalize. So in other words, packing the protein, repacking the protein inside. So that was a very important function. Now from that we went to a study of how the chaperones all lined up. Now this is a complex, very busy slide, so you're not to look at this seriously, looking at trying to look at the amino acids. But what you have to look at is that as these chaperones are lined up, from the top are hyperthermophiles, at the bottom are cyclophiles. What you see is that the core of the protein from here to here at the end is highly, highly conserved. That in some cases, in many cases, the entire blocks of the same residue are present. But you also see that here in the C-terminus, in the black ring, you see that everything starts to get very, very unconserved, and there's divergence here. And my student Iben Luo looked at this in terms of what happened at the C-terminus. So you blew this up now. First of all, there's highly conserved in all of these species with temperatures of growth from 23 to 110. And what you see is that as you go up in temperature, you also go up in the amount of charged residues in you. So our pyrococcus is here, and it has 10 residues that are charged, and many of them are positively charged. So Iben Luo showed here had the idea to actually change these residues systematically to non-charged residues. So here he changed one glutamate into a glycine, and then second, and then so on, until he got one where it's called MA, mutated all of the charged residues, the negatively charged residues. And when he did that, he found that actually the optimum temperature for the chaperone could be tuned down. So as he tuned it down, the mute ore came down to 40 degrees. So we could actually change the temperature range for this whole chaperone by changing the number of charged residues. So it looked like this was a little switch, a variable switch like you have on your light that you turn up or down for different brightness, but it was affecting the stability. Here we see the stability at 50 degrees. We find out that the ones that are at without any residues, they are extremely good. But if you look at the wild type, that actually there's no difference or no difference between wild type and the GDH, which has been used as a folding substrate now, is completely preserved here with a low temperature chaperone, but it's not preserved at all, hardly at all, with the wild type. So it's also good for folding proteins. We also found out that you could take the C-terminus, the little tag on the end, and put that onto the body of a cyclophilic chaperone from the MB chaperone and put it into a new mode. In other words, you could switch the thermostability up, and that finally got us to publish this work, because if you just mutate things and the temperature and stability and function goes down, people are not impressed until you can make it go up as well. So we could find out that the cyclophilic chaperone, after you switched it on with a C-terminus from the hyperthermophile, is actually going up in temperature, in its temperature of activity and its temperature also of function. So that gave us an idea to use the chaperone for another medical experiment. It happens that medical genetics is looking at a large number of diseases, and some of them are in chaperone. And this is a disease which is in the CCT5, the number 5 subunit of the chaperoning from humans. It's discovered in a Moroccan family where 4 siblings had a terrible disease. They had mutilating neuropathy from 1 to 5 years old. They couldn't feel their legs particularly, and then particularly in the feet. And 2 of them had deep ulcers, and one, the most badly affected one, had to have both legs amputated. Discovered that this was single amino acid replacement, histidine changing into arginine at position 138. And this publication was really our starting point. We looked at the human CCT5, and we modeled it on our Pyrococcus chaperone. And this was me moving into the medical school, trying to find out how to do neurosurgery. This is a cartoon which says, yeah, Frank's neurosurgery works, $200. And I adapted, in other words. If you're an extremophile person, you have to be adaptable. So this was me adapting to life in medical school. So we eventually looked at the CCT5 mutation. We modeled that into the protein of the Pyrococcus, and found out that you could actually place it in a position within the folding cycle. And this paper became a model for not only this, but other neuropathies as well. So what we did, as you can imagine, we took the model, and here's the actual site of the mutation. It doesn't give away anything as to what it's doing in the structure and catalytic cycle. However, it has a big effect on the catalysis of the chaperone. We put this, first of all, we had the Pyrococcus furiosus wild type. What we used, as you could imagine, was one of the destabilized variants of the Pyrococcus, so we didn't have to do all of our work at 100 degrees. We changed the site to a histidine, which was the original site in the human version, humanized. And then we changed that site also to arginine, which is the amino acid which is in the mutation. What we found was this is the ATPase activity of these complexes. There's the wild type and the humanized have very similar ATPase profile, around 40 degrees maximum. However, there's very little ATPase activity in the mutated one. Wang Min, Korean guy who's my postdoc during this time, he showed that by taking the complexes from these and putting them on a native polyacrylamide gel, first of all, you can see the open and closed forms on these gels. Here's a diagram of what functions. The open form is like skydiving, if you want to think about it like that. It's skydiving in the open form is slower than in the closed form. What he found that the quite similar structures between the wild type, the humanized, but when you came to here, you found very little of the 16 mer. In fact, most of these were down in dimers and monomers. And this shows the SDS gel showing that we loaded equal amounts on each of these layers. Then Dario Squiglon, Italian, who spent a year in my lab, and during that time he was very productive. He took these three and he did scanning calorimetry with the three of them. What he found was that they all, interestingly, had a TM shown here, very similar. And this is the temperature at which the monomer, the subunits, start to break down the structure. So the monomer structure is not affected by the mutation. It all happens. The final melt of this protein happens at 102, 103 degrees. However, there's a transition further on down here where the protein, we believe, falls apart and disassembles. And so the protein that's in R here has a very big transition at 90 degrees, which falls apart, and so it's not able to maintain the complex. Okay, so then we did concentrations of different concentrations, and these allow us to look at the whether this is an equilibrium or non-equilibrium, and it turns out to be a non-equilibrium transition. And what happens was that it actually gets irreversibly disassembled, and actually all of them are common to this. And this is the ATP binding. What we found out was that actually at ATP is bound to the humanized and to the R quite similarly, but the wild type had a different binding protein. We still don't fully understand why the humanized is different from the ATP binding to the wild type. And here we looked at binding in the isothermal titration calorimeter. Interestingly, it shows that there's a stoichiometric binding of 16 ATP molecules to the complex, and that this is an endothermic binding for the H and the CD. However, the R, it's an exothermic binding, which is the other way around, not expected. And that gives some idea of why the ATP hydrolysis is so low. Actually, its binding is a very low affinity. Just going back here, what you see is that the difference between this complex and this complex, you have eight subunits here, which are all different. One of them is the bad one. It's wrong, and it has a profound effect on the complex, and a profound effect, a very bad effect on the humans that carry this mutation. However, in the Pyrococcus, you have 8 subunits, and they're each carrying the mutation, whether it's good or bad. So this biochemically amplifies the effect of the mutation 8 times. So for this complex to be effective at all the mutations that are in humans have to be very mild. They have to be hardly noticeable. However, when you put them into this, where the effect is amplified 8 times, you can start to notice them. Another study by a group at MIT found that they couldn't see any difference between the mutated version of this and the unmutated version. However, we can see the difference here when we amplify it. So it shows some of the uses to which archaea can be put, because they have simplified complexes compared with higher eukaryotes. So again, Pyrococcus is good for that. Finally, this is the tree of the chaperones. Chaperones. What we see is a group here, which is coming out of the side. Here are the groups which are the group 1, the ones that are also in the bacteria. Here are the ones that are in the group 2 in humans, and here's a group which is different again. And so we've concentrated on this group, and it turns out to have been very interesting, because somehow this group is really at the base of the phylogenetic tree of these chaperones. So maybe this is close to where these two groups, which clearly belong in the same ancestry, split apart. So these could be remnants of their ancestors hanging out in different really strange bacteria. But they all have the same architecture, the same apical, intermediate, and equatorial domain. And what we did then is to look at this principle. This is a little complicated slide, but it shows you some of the complexities that are in human chaperone pathways. What you are shown here on the left, where this is a defective chaperone, and on the right is the effective chaperone, the wild type. What you see is that there are defects in the pathway, the schematic pathway from binding, hydrolysis, etc. And so what's affected is not only the ability to break down ATP, it's also the ability to bind cofactors, because these chaperones do not act by themselves. They are interacting with a large number of other chaperones. And each time there's a defect, it is reflected in the amount of interaction and the nature of the interaction with the other chaperones. So this is a study that co-workers have just published, and it's a very interesting analysis of how we should look for the next layer of function for the chaperone. Finally, I want to talk a little bit about the area of archaea that has not been really touched so far. All the omics fields have been applied to the archaea, metagenomics, genomics, transcriptomics, proteomics. What is very interesting, though, is what is the status of a living cell as shown by the metabolome, the small molecules that are inside it. And so Kia Hagiwara, who's my postdoc, she's just now got to a permanent position at the NIST, the National Institute of Science and Technology. She did a study with a hyperthermophile to see what the difference between growing it on starch and carbon monoxide. So quite an interesting organism. It grows by breaking down starch, or else it can grow by breaking down carbon monoxide, making CO2 and hydrogen. And it's growing differently here is the principal components analysis, showing that actually there are two components shown by different groups of metabolites. And her modus operandi, our approach was to use a very high field NMR. This is the largest NMR at the lab she's at, and to visualize as many metabolites and as she could. And this is a bit like doing proteomics. We still don't have all of the metabolites, obviously, but we have enough to show that there is a different profile. And that's the beginning of I believe, a new growth field in archaea, where if you're wondering about what's the next thing that needs to be done, I think metabolomics is going to be a very fruitful field. So summarizing what we've said so far, we looked at the physical challenges. We looked at the different areas that allow organisms to grow just at the edge of their survival. We looked at hyperdiscovery, the discovery of hyperthermophiles, hyperacidophiles. We looked at some of the physical adaptations to physical challenges like high pressure. We looked at two medical applications of research in hyperthermophiles, vaccines and genetic disease. And then we just touched on the next frontier, which I believe is going to be metabolomics. Just wanted to, this is my lab some years ago. These people have mostly gone on to fruitful careers in Germany and medical school, and back to a pharma company. This is Dario, a pharma company in Switzerland. We are still working together with Sanshin Cha and in Seoul, in Korea, and Francesco Capello in Italy. And just want to say thank you to NASA and the PATH Foundation and NSF more recently for funding and support from NIST for Kiha, her current job and support. And thank you for your attention.
Thank you very much for your speech, Dr. Frank. We are now going to proceed with a few questions from the public. Damos paso a una ronda de preguntas. Nuestro compañero de comité, Felipe Gatica, estará compilando las preguntas para dejárselas al Dr. Frank. Felipe?
Aquí estoy. Good afternoon, dear Frank.
Yeah,
Do you hear me well?
I hear fine.
Okay, so let's go. Maximiliano Amenabar says in the chat, thank you for your great talk. I would like to ask you, what is known about chaperones, phylogeny, and the evolution? Were chaperones likely present in Luca, considering that Luca was likely thermophile in nature? Thermophilic in nature.
I think almost certainly chaperones were present in the Luca. It turns out that although some proteins can fold themselves, but any sort of complexity and recovery and salvage of proteins when they unfold is almost always achieved by chaperones of one sort or another. Chaperones, we're more interested in chaperones because they're very complicated, and so the Luca is a real debate whether the Luca would have a chaperonin, which is the complex of a 7 or 8 membered ring. We don't, obviously nobody knows, but it looks like the split between the group 1 and the group 2 for chaperonins has happened very early on between the time of bacteria and the time of archaea, and certainly before eukaryotes appear.
Okay, thank you for your response. Well, Maximiliano says thanks in the chat too. Ignacio Lopez says, thank you very much for your presentation. It was very fascinating topic. About vaccines, how do you think it could scale to a massive level?
Yes and no. The application to vaccines is at this point for a particular case of the live attenuated vaccines. In some ways, these are very useful because you can actually drink them. You don't have to inject them, and they can obviously be used in developing countries. They are being used now, and they also can be relatively cheaply produced. But every time you add another protein to a vaccine, you add some complexity to the regulations governing the getting a new vaccine into circulation. The approval process is usually a couple of million or many $1 million of going through the safety checks and so on. And for doing this, we discovered as we got our vaccine with a new protein from a extremophile in it, we tried to see how this would go through that. We have to first of all raise the funds in order to go through the regulatory process. And so it's adding an extra few millions to the regulation hurdles you have to go through. So the short answer is yes, like all therapeutic strategies, it's possible, but usually you need to actually raise pretty big funds in order to go through, and you have to usually be in the corporate world. And Anna Baum, who gave the first talk, gave an idea of the things they can do, but they're actually in a company, and the company is not in an academic lab. So they're doing things with monoclonal antibodies that would be impossible to do in an academic lab. Same thing with vaccines.
Well, thanks for your response. Joaquin Atala is asking, here's a question for Dr. Robb. if you understood correctly, the HSP you use for vaccine stabilization were used in vivo in attenuated strains. Is it possible you use the HSP to stabilize single proteins in vitro?
Oh yes, indeed. We've done this a lot in experiments and also in terms of keeping proteins active and able to maintain at room temperature. So yes, we could stabilize subunit vaccines as well. Nobody has, as far as I know, nobody has done that. So there is some interest in it, and we have the chaperones for it.
Okay, thank you. Well, Maximiliano Urrutia says, "Hi, Dr. Robb. Thank you for your participating in this Congress, giving us a very illustrative lecture. Besides of the use of thermophilic chaperones in developing temperature-stable vaccines, which other biotechnological approaches using extremophiles are currently being proposed or studied?"
Well, the biotechnology applications are happening in molecular biology a lot. They have many components that we use in the lab are derived from extremophiles of one sort or another, the restriction enzymes, the polymerases, etc.
Yeah, Techman.
So there are plenty of those. We also, part of my talk I didn't go into was the use of enzymes for biofuels production. Some of the enzymes are, for example, breaking down lignocellulose for making lignocellulose fermentation. Those are very interesting. At the moment, actually, the whole biofuels field is very down because of the low price of oil. However, a couple of years ago, there was interest, and we had a paper in Nature on a hyperthermophilic cellulose. And that's still sitting out there, waiting for the price of oil to go up. What other many of the labs of Dr. Jenny Blamey are doing many different enzymes, commercial enzymes and commodity enzymes are, of course, a very, very big worldwide feature, and especially enzymes that are stable and still active are very useful for and profitable. So they've been found to be effective in many applications down the years.
Well, okay. In fact, our dear Dr. Jenny Blamey is asking in the chat of Zoom. So she says, thinking in biotech applications, can you comment on the combination of using enzymes for extremophiles together with HSP from extremophiles other than in molecular biology?
Well, actually, I think there's a feature of doing this. You could imagine recycling enzymes, even immobilized enzymes, which were then restored to activity by refolding them. I actually have a history of doing that, proposing that to large biochemical enzyme producing company. I won't name them, but I proposed that this would be able to, they could market the chaperone along with the enzyme that they're selling. And then the person could actually reuse the enzyme or stabilize it for a long period of time. So unfortunately, I'm not a very good business person, I think, because the biotech companies make their money because you buy an enzyme from them, and then after 6 months or a year, the enzyme is dead. You have to go and buy another enzyme, another shipment. So that's where they make their money. If you sold enzymes that would never go bad, then that profit stream would not be active. So we went a long way. We had wonderful results. Came to the end to talk to the committee that actually looks at the profit and loss, and they said no, we're not interested because it's not going to earn us money the way we earn money, which is to sell unstable enzymes and wait for them to go bad. So yeah, there's still hope, but you have to think about the commercial strategy very, very hard before you do that.
Okay. Well, we have another question from Maximiliano Amenabar. So he says, it has been recently suggested that RNA can function as a molecular chaperone for RNA folding, which might support the idea of the RNA world hypothesis. What is known about nucleic acids acting as chaperones? Is this a common strategy used by organisms?
Yeah, it is actually. When you think about the structure of the ribosome, it's self-catalytic and self-chaperoning. The ribosomal assembly, and so there's a lot of RNA chaperoning going on in there. I can't comment on the RNA world being supported by this. Obviously, there are catalytic RNAs, but their range of activities are quite low. They're not generally in central metabolism or other areas where essential catalysis would take place. So whether the RNA world had RNA chaperones, I don't know. It's an interesting speculation.
Well, okay. This is a question that in the beginning of the talk, I began to ask. So, how this area of research has developed over the years?
Well, trying to give some idea of the progression. First of all, the very existence of these organisms near the survival limits of extreme conditions was unknown. So first of all, there was a wonderful discovery era, where a lot of strains were discovered. Now it's pretty routine that organisms can grow above 100 degrees. Catalysis and even industrial processes above 80 degrees are pretty common. So we're entering a new era. Historically, it was really the microbiologist era. Now it's microbial genetics and biochemistry and systems biology that's important for the field to progress further. The frontier, much of the frontiers now are with the people who can interpret big data sequencing, because there are so many virtual organisms there defined just by their assembled genomes, and there's no isolation. And so that is now that there are many organisms we can't grow, but we know they exist because the genomes exist. And to get at their functions, there's going to be very large scale protein expression, protein assays, protein discovery, because many of these genomes have genes that aren't known, and they're not homologous to known genes. And so what their proteins do is mysterious. So there's still a lot of discovery going on, but it's at the level not of microbiology, but of molecular biology and biochemistry.
Okay, thanks. Great. Well, I think that this will be the last one. So, Diego Ignacio Jesus Salas says, have your laboratory also studied the characteristics of small chaperones in extremophiles, like the HSP70 families?
Actually, HSP70 does not exist in extreme thermophiles. So that's a problem. So one diagram I showed was how many classes of chaperones are not existing, not represented in high temperature archaea. So HSP70s are interesting, but they so far are kind of non-starters for extreme thermophiles. We have interesting data for non-extreme thermophiles, but the classes of chaperone that people have looked at are mainly HSP60, small heat shock proteins, the AAA plus ATPases, and in some cases HSP70s in lower temperature organisms.
Well, okay. Thank you for your time. I'm leaving, but first I want to give some words to Jenny Blamey and Maximiliano. La tiré en español. Quería dar unas gracias especiales a nuestra doctora Jenny, como comentaba, y al doctor Maximiliano Amenábar, porque ellos nos ayudaron muchísimo para poder estar aquí con nuestro querido Frank Robb. Así que realmente muchísimas gracias. Como mucha gente sabe, el congreso se realizó en un mes, así que fue una carrera con todo el tiempo, así que muchísimas gracias. Well, Frank, I'm leaving. It's nice to meet you again, to see you again, and we will be in touch. Okay. Goodbye.
Thank you so much for the opportunity and the invitation. I really enjoyed this.
Great.
Thank you, Dr. Frank, for your speech. We would like to ask a question from the staff. If we are making this transmission online in YouTube, we would like to know if this transmission can stay like a video on YouTube.
Yeah, I give permission for that, no problem.
Thank you very much. We appreciate a lot.
Okay.
I don't know, Dr. Frank, if you would like to say a few words of
well, this is the end of the conference, I think. So I just wanted to say what a great challenge it has been, and what a refreshing time it's been to see a whole week-long conference with a good humanitarian cause, and also bringing together so many different people with the normal COVID isolation trials and tribulations. So it's been refreshing to be associated with this, and I thank you so much. know those people who have had different experiences in the COVID crisis. It's helpful. I myself, it's time at home. Right now, I have to get a test before I can go back to my lab. Medical school insists on that. So we all have had different trials, and we look forward to a time when we can get back to normal. But in the meantime, I really admire you and salute you for doing this in a way of progressing scientifically, even despite the challenges. I wish you luck for the follow-up, and I hope that things can be different next year. but meanwhile, well done. Very well done.
Thank you very much for your words. Quisiéramos dar un espacio para invitar a Wilson Castillo, presidente de la comisión organizadora del 36º Congreso Nacional de Estudiantes de Bioquímica. Wilson, sí. ¿Estás ahí?
¿Sí me escuchan?
¿Te escuchamos?
Bueno, ya me presentaron. Soy Wilson Castillo y antes de comenzar me gustaría mostrarles un video que me permiten compartir. Creo que ya puedo. Sí. Espérenme. Deberían ver agosto 2019, ¿me confirman?
Te confirmó, se ve.
Perfecto. Ahí, espérenme. Se me olvidó poner alguna cosa. Ahora sí. Ahora deberían haber agosto 2019 de nuevo. ¿Sí?
Sí.
Ya.
Cada año se lleva a cabo un congreso donde una de las 11 filiales se prepara para recibir a todos los estudiantes. El año 2020 se llevará a cabo el 37º Congreso Nacional de Estudiantes de Bioquímica en el campus Eloísa Díaz de la Universidad de Chile, donde reside nuestra facultad, la Facultad de Ciencias Químicas y Farmacéuticas.
Por quinto día consecutivo se registraron incidentes en cerca de 15 estaciones del metro de Santiago de Chile, luego de la última subida del precio de los tiquetes.
Hoy, ejerciendo mis facultades constitucionales, he decretado estado de excepción constitucional de catástrofe en todo el territorio nacional.
Santiago de Chile amaneció fuertemente militarizado luego de que el presidente Sebastián Piñera activara la Ley de Seguridad del Estado.
Cacerolazos que se sintieron incluso en la isla de Pascua.
Puta hermano, ¿sabéis qué? Estoy preocupado por todo esto del estallido social. No sé si vamos a poder hacer el congreso el próximo año.
Nah, yo cacho que hay que esperar nomás. Yo cacho que entre diciembre y enero esto se calma para marzo poder trabajar tranquilos.
Puta, sí. Ojalá tengáis razón. Hay que esperar nomás.
Estamos en guerra contra un enemigo poderoso, implacable, que no respeta a nada ni a nadie.
Prometía ser la marcha más grande de Chile y si bien aún no están las cifras definitivas, a esta hora ya lo fue.
Interrumpimos nuestras transmisiones habituales para dar cuenta de la información del minuto que tiene que ver con la confirmación del Ministerio de Salud del primer caso de coronavirus en nuestro país.
Pucha, hermano, estoy preocupado porque ahora en marzo viene el estallido social 2.0 y llegó el coronavirus acá a Chile, loco. Está brígida la cuestión.
Sí, hermano, está acuática la cosa. Pero mira, tómalo con calma, no nos preocupemos. Si llegan a cancelar el Lola, ya ahí recién nos preocupamos. Cachemos qué onda.
De último minuto, Lollapalooza acaba de suspender el retiro de pulseras. Ojo con la información que estamos dando.
El Ministerio de Salud está evaluando suspender los eventos internacionales que va a venir una gran cantidad de gente, entre ellos el Lollapalooza.
La medida más severa que debo anunciar por instrucción del presidente de la República es una cuarentena total en el Gran Santiago.
Say something, I'm giving up on you. I'll be the one if you want me to. Anywhere I would have followed you.
Bueno. ¿Me escuchan, cierto?
¿Escuchamos?
Perfecto. Bueno, primero decir muchas gracias por la oportunidad que nos dan de poder promover el congreso del próximo año. Primeramente quiero felicitar a todo el equipo que logró sacar adelante este gran proyecto del congreso online, el cual servirá de ayuda para muchas personas. De parte de la comisión organizadora del 37º Congreso Nacional de Estudiantes de Bioquímica y Carreras Afines, queremos enviar todo nuestro apoyo a todos los presentes y a sus familias. Ha sido un año muy difícil y aunque se ha visto complicado para muchos, nos alegra saber que la comunidad ANEP sigue acompañándonos. Como comisión, hemos tenido muchos problemas para sacar adelante el 37º Congreso. Sin embargo, estamos activos para brindarles una experiencia inolvidable para el 2021. No me queda mucho más que dejarlos invitados a estar atentos a nuestras redes sociales, en donde pronto subiremos información de vital importancia para lo que será el 37º Congreso Nacional de Estudiantes de Bioquímica y Carreras Afines en la Universidad de Chile. Se vienen muchas cosas nuevas y estamos emocionados por hacerlas realidad. De parte de toda la comisión, les mando un abrazo grande a lo lejos y espero que nos podamos encontrar pronto. Nos vemos en el congreso del próximo año y felicidades por todo lo que están haciendo en los H. Muchas gracias.
Muchas gracias, Wilson, por tus palabras. Para dar el discurso de cierre, queremos invitar a uno de los corazones de este proyecto, quien hace aproximadamente 2 meses me invita a formar parte de un pequeño proyecto de divulgación científica enfocado más que nada a nivel de nuestra carrera. Pucha, le dije honestamente, la divulgación científica no me llama en este contexto, pero te ayudaré en todo lo que pueda porque eso hacen los amigos, ¿no? Hoy siento la necesidad de retractarme de mis palabras de aquel entonces y estar totalmente agradecido de haber formado parte de este proyecto, comprender la suma importancia de ser parte de la divulgación científica en plena crisis sanitaria, haber aprendido más de astrobiología, la disciplina que más me apasiona, e inclusive, ¿por qué no decirlo?, todo lo que aprendí de edición audiovisual, por más horrible que haya sido. Netamente, para estar a la altura de una labor a la que quiero creer, tuviste toda la confianza en mí para decir, yo quiero que Pablo haga de anfitrión. En los tiempos en que nunca se nos pasó por la mente preguntarnos quién iba a estar allí adelante. Por eso, y por mucho más, Víctor tiene mi enorme agradecimiento. Espero poder hablar en nombre de todo este comité al decir que desempeñar este congreso fue sin duda un arduo trabajo, pero a la vez fue una bonita experiencia de la cual formar parte y compartir con todo un equipo súper admirable. Pero quiero creer que este equipo se vio guiado por un liderazgo único en ti, que no hubiese querido que fuese de otra manera. Y tal como hace 2 años, hoy me lleno de orgullo al invitarte nuevamente a este escenario virtual para dar este discurso de cierre. Quiero darle la bienvenida a Andrés Masilla.
Hola, ¿se escucha?
Te escuchamos.
Gracias, Pablo, por la introducción. No me esperaba eso, en verdad. Ya voy a partir. Esto va a salir un poco improvisado y a la vez no. Tengo un par de apuntes también, pero bueno, esto va a ser un poco difícil porque este es mi último año, entonces es el último año para varias de nosotras que estamos en la comisión. Entonces voy a partir. Estimadas autoridades, compañeros, socios de la asociación y futuros colegas asistentes a esta versión del Congreso Online. Hace un mes, exactamente a las 11 de la noche, conversé con Patricia, o como le decimos sus amiguis, la jefa, acerca de elaborar esta nueva versión de un congreso de estudiantes modificándose a una versión online. Esto partió simplemente como una idea chica, 3 días, y que fuese un ciclo más de charlas, pero con actividades variadas y similar a un congreso de aquellos que suele realizar la asociación a la cual represento. Sin embargo, esta idea no pudimos realizarla en solitario. El mismo día le hablé a compañeros y amigues a los cuales realizamos la 35ª versión del Congreso Nacional de Estudiantes de Bioquímica y Carreras Afines, justamente y también realizada en la Universidad de Santiago. Todos quisieron apañar esta inmensa idea sin ningún pero encima, solo su buena disposición y cariño que nos teníamos entre nosotros. Esa misma noche creamos un grupo, propusimos ideas y se conversó de que esta actividad ya no sea solo para estudiantes de Bioquímica USACH. ¿Por qué no escalar y hacer algo mucho más ambicioso? Pues entonces decidimos agrandarlo, que ya no fuese solo un par de días, sino que tomamos la drástica decisión de hacer que este enorme proyecto de divulgación científica sean 6 días, y que por lo demás nos propusimos hacer algo sumamente imposible de hacer, que es conseguir todo lo propuesto para realizar este nuevo congreso en tan solo un mes. Rápidamente empezamos a buscar gente de otras generaciones porque la mayoría de los que en este momento estábamos realizando la actividad nos encontramos en tesis u otro tipo de actividades académicas y contábamos con muy poco tiempo para su dedicación. Pero muchos estudiantes quisieron sumarse a esta iniciativa. Además de esto, quisimos agregar nuestra cuota de apoyo al pueblo chileno a través de una campaña de beneficencia, como le decimos, y de esta manera colaborar a distintas organizaciones que se encuentran apoyando mediante ollas comunes o canastas familiares a familias que en este momento de pandemia se encuentran pasando por situaciones difíciles. De esta manera organizamos esta primera versión de un congreso online de bioquímica, una actividad sumamente enriquecedora debido a la calidad y la cantidad de temas que les mostramos a ustedes. Como estudiantes de bioquímica, y es más, como personas, encontramos sumamente necesario el brindar un apoyo a la comunidad, y qué mejor que haciéndolo a través de algo que sabemos hacer, un congreso online, que divulgue ciencia de manera totalmente gratuita. Estas palabras de agradecimiento también son para la decana de la Facultad de Química y Biología, doctora Leonora Mendoza, por su apoyo constante y sus palabras de ánimo cuando atravesamos complicaciones. La vicedecana de docencia, doctora Brenda Modak, y nuestra jefa de carrera, la doctora Marcela Wilkens, cuya disposición para todas nuestras solicitudes siempre fue muy bien acogida, y nos ayudó con todas las funciones de inclusión y apoyo. Y también el soporte de internet, contactos dentro de la universidad, entre muchas otras cosas. Voy a pedir a la comisión organizadora que se muestre en la pantalla, porque ahora me voy a emocionar un poquito. A los más importantes, quiero agradecer a todos mis compañeros y amigues que ayudaron en toda la organización de este evento, a todas aquellas que en este momento están mostrándose por las pantallas de su computador porque los estoy obligando a mostrarse. Porque sin ustedes esta idea que partió como una simple semana de charlas, terminó siendo un evento enorme, que abarca más de 500 estudiantes de bioquímica de todo Chile, más de 700 estudiantes de las ciencias exactas en todo Chile, e incluso con estudiantes de países como España, México, Venezuela, que pudieron asistir a este precioso evento que hicimos con mucho esfuerzo y dedicación. Agradecer a las personas integrantes de la comisión académica, a Úrsula, a Fabián, a Jorge y a Felipe por haber generado los contactos con cada uno de los expositores, que presentó y participó en las actividades como talleres, foros y simposios. También agradecer enormemente a la Comisión de Difusión, a Dafne, a Camila Retamal, a Carolina Cortés, Francisca Tapia, a Ignacia González, Tamara Mateluna y Carla Contreras por los maravillosos afiches, el libro, el manejo de las redes sociales y la ayuda en la promoción de los eventos mediante nuestras plataformas digitales. A la Comisión de ODEP, a Romina, al Nico, a la Camila Sandoval, a Rodrigo Sañartu, Victoria Flores, a Francisca Grene, Ignacio López y Diego Barrenechea, por la cuota de entretención durante la semana y por la organización de los distintos concursos y actividades deportivas online que se realizaron y se van a realizar el día de mañana y el día domingo. También agradecer a Camila Sagredo, a Pablo Jiménez y a José Benavides por su ayuda en la Comisión de Beneficencia realizando los contactos con las distintas campañas y los sectores de Puente Alto, El Bosque y también de Peñalolén. Por último, agregar a la Comisión de Logística, Gladys Serrano, a Patricia, o como le decimos la jefa, y a Paula Leighton por manejar las planillas, realizar los formularios de inscripción y manejar los correos para mantener todo un orden durante la planificación de este congreso. No me cabe la menor duda de que ustedes serán grandes profesionales con ambiciones sumamente fuertes y que llegarán a donde planeen estar, porque son grandes de corazón y también sumamente grandes como compañeros de trabajo. No quiero dejar afuera de estos agradecimientos a Manuel Narbona, estudiante de periodismo de la Pontificia Universidad Católica de Valparaíso y miembro de Nuevo Foco, que nos ayudó a moderar el foro El auge de las fake news y también ayudó a la Comisión de Difusión. Ya. Actualmente nos encontramos sobrellevando una de las peores crisis, tanto ambientales como económicas, y peor aún en situaciones de salud. Nos encontramos atravesando una crisis sanitaria sumamente importante, donde Chile se ha posicionado dentro de los peores países que ha enfrentado este proceso, que sin duda ha sido doloroso en muchos sentidos para nosotros. Sin embargo, de los errores debemos aprender y sacar lecciones de todas estas situaciones para que en el futuro sepamos cómo y de qué manera debemos enfrentar de una manera más óptima cualquier eventualidad. Porque aquí nos dimos cuenta que la historia no es solo algo que nos muestran durante la educación media y que debe ser desechada, y que por lo demás no debiese ser quitada de la malla educacional de la enseñanza media. Esto nos demuestra lo importante que es informarnos, aprender y sobre todo adquirir una educación científica de calidad para formar personas con conocimientos que sepan a qué nos estamos enfrentando el día de mañana, que puedan divulgar, interesarse y enamorarse de la ciencia. Es importante que exista este cambio en la educación de una manera urgente, para que el día de mañana seamos ciudadanos con pensamiento crítico y que podamos ser capaces de realizar cambios a futuro para la sociedad. Agregar a lo expresado anteriormente que es necesario dejar de crear barreras y sectores científicos que se encuentran dividiéndose. El día de hoy más que nunca necesitamos una comunidad científica consolidada y que sea crítica de lo verídico y no caer en los amiguismos. Aceptar que podemos cambiar de opinión y errar en cosas del pasado, porque toda nuestra educación nos hemos enseñado a nosotros mismos acerca de la tolerancia a la frustración y aceptar que no hay competencias entre nosotros, pero cuando llegamos al momento de decidir quién tiene la verdad o no, apuntamos con el dedo y cedemos hacia el lugar que más nos privilegie. Dejemos de lado estas malas prácticas entre aquellos que están insertos en la academia y quienes buscaron opciones laborales distintas a la investigación. Aprendamos a dejar de ser tan ególatras por ser científicos y dejemos nuestros privilegios de lado y hagamos una ciencia más humana y sin barreras. Sabemos que en la actualidad la ciencia y el desarrollo son puntos sumamente importantes para avanzar como sociedad y progresar como país. Sin embargo, Chile es uno de los países de Latinoamérica que menos invierte en ciencia y tecnología, por lo que el bajo apoyo económico nos limita a buscar otras posibilidades de aplicación y desarrollo investigativo e ingeniar de qué manera podemos realizar ciencia. Esto sumado a la precarización laboral a la que nos vemos sometidos algunos estudiantes por la falta de oportunidades laborales una vez egresados. Para progresar como país, debemos atender las demandas desde todos los lados, y como científicos necesitamos que nuestras demandas sean escuchadas por un ministerio consciente de nuestros problemas. No sirve imitar a países desarrollados y creerse uno de ellos, si lo que realmente necesita el país para ser desarrollado es invertir en ciencia, porque detrás de cada desarrollo hay un avance científico. Deberían sentir vergüenza las autoridades del gobierno que se llenaron de flores cuando en una pandemia se ha demostrado la incompetencia de estas materias en vez de escuchar a quienes realmente hubieran hecho un trabajo mejor. Que ojalá no olviden todas las necesidades que tiene el mundo científico, porque ahora piden y dan, claramente por la angustia que tienen, pero antes no nos daban más que migajas. Es por esto que no es difícil concluir que la visión del científico encerrado en 4 paredes, absorto de la realidad que lo rodea y con el único propósito de sacar más y más papers, está totalmente obsoleta. Hoy en día, si queremos cambiar realmente las cosas, necesitamos sacar un rato al científico de su silla de laboratorio y que entre en la palestra de lo político y lo social, de hacernos escuchar y hacernos valer como los engranajes vitales para que un país funcione siempre hacia adelante, porque el motor del conocimiento, al fin y al cabo, es el único que nos sirve para encontrar las reales soluciones a los problemas que necesitamos para avanzar como país. Sin el mundo de las ciencias, las humanidades y las artes, un país no es más que un generador de dinero que solo seguirá favoreciendo a los mismos, al menos que nosotros salgamos de esa inercia e impongamos nuestra voz para cambiar las cosas. Quiero finalizar entregando mis mayores agradecimientos a ustedes por su asistencia a todas las distintas instancias que realizamos como comisión organizadora para que puedan nutrirse de conocimiento, aprender y divulgar algo que nos apasiona. Muchísimas gracias por todo su apoyo y muestras de cariño expresadas a través de cada mensaje de agradecimiento por las plataformas virtuales donde realizamos este evento. Y también por colaborar en la campaña de beneficencia que realizamos para ayudar a muchas familias de la región metropolitana. De manera personal, agradecer a todos mis amigos por dar un espacio de su tiempo en nuestras juntas como comisión, en las rabias, las prácticas, las risas y su apoyo constante para sacar este enorme proyecto. Les agradezco enormemente por apoyarme en idear y moldear esta enorme propuesta, ya que para algunos de nosotros es nuestro último año como estudiantes de pregrado. Sin embargo, nuestras ganas de luchar por nuestros derechos como científicos y como personas nos van a mantener siempre unidos. Me despido, y por último, como comisión organizadora, SIPO, aprobamos nueva constitución.
Damos las gracias a Víctor por sus palabras. Agradecemos a los miembros del comité que se hicieron presentes. Y despedimos así este primer Congreso Nacional de Estudiantes de Bioquímica y Carreras Afines. Les damos las gracias a todos y todas por su participación, esperando que hayan aprovechado al máximo cada una de las actividades. Esperamos que todos los conocimientos aprendidos puedan ser puestos en práctica en el tiempo y compartidos con el mayor número de personas en sus círculos cercanos, que logren adentrarse con su interés en todos los campos que deseamos abordar en esta instancia, y quizá los que tampoco pudimos: inmunología, microbiología, biotecnología, astrobiología, entre otros, sin dejar de lado la importancia de la divulgación científica. Esperamos haber sido una puerta de aprendizaje, pero quizá también haber sido una ventana de distracción o alivio ante todo lo difícil que ha sido este año para todos. Esperamos que nuestros socios disfruten al máximo las Olimpiadas Virtuales de Bioquímica, planeadas con mucho cariño durante este fin de semana. Es un honor, como anfitrión, dar por finalizado el primer Congreso Virtual de Estudiantes de Bioquímica y Carreras Afines. Que tengan todos un excelente fin de semana y esperamos volver a vernos pronto en la Universidad de Chile o donde podamos vernos. Muchas gracias y nos estamos viendo.